Today, we're diving into something pretty amazing. We're going to see how scientists take one of nature's tiniest tools, these things called enzymes, and blow them up to an industrial level. This is literally the story of turning microscopic engines into manufacturing might.
But let's start with a big idea. Just think about this. What if the future of making medicines wasn't all about harsh chemicals, but about biology? What if we could actually harness nature's own microscopic engines to create the drugs we all rely on, but in a much cleaner, greener way? That's the huge idea we're going to unpack right now.
So, to even begin to answer that question, you and I first need to get to know the stars of our show, nature's tiny engines.
So what in the world is an enzyme? Well, to put it simply, it's a special kind of protein that acts as a biological catalyst. You can think of it like a microscopic matchmaker or an accelerator. It finds a very specific chemical reaction and just makes it happen, sometimes millions of times faster than it would on its own. It does all this by basically lowering the energy barrier to get things started.
And this visual right here is the perfect way to understand it. It really is like a lock and key. The enzyme, that's our lock. It has this perfectly shaped little pocket, and only one specific molecule, the key, can fit inside. Once it slides in, boom, the enzyme gets to work, transforms it, and then releases a totally new product. It's just unbelievably precise and super, super efficient. And you know, this isn't just some cool science fair project. We're talking big business. The global market for industrial enzymes is expected to hit over $11 billion by 2030. These tiny little engines are already powering huge industries, from the food you eat to the medicines you take. Okay, but here's the catch. You can't just scoop an enzyme out of nature and expect it to work in a giant factory. It's not that simple. We've got to unlock its full potential. And, well, that comes with some pretty serious hurdles. And this slide, it just lays out the central problem perfectly. See, in their natural state, enzymes can be kind of fragile and unstable. They're a pain to separate from the stuff they make, which means you can't really reuse them. But industry needs the exact opposite. They need enzymes that are tough, stable, easy to recycle, and that just work at peak performance 24/7. So how do we get the huge amounts of enzymes that we actually need? The answer is a process called fermentation. Scientists pick out specific microorganisms, like the bacteria and fungi you're seeing here, and they basically turn them into tiny living factories. By giving them the right genetic instructions, these little microbes can be programmed to pump out massive quantities of the one enzyme we want. Now, let's meet the hero of our story today and see how this all clicks together. We're going to zoom in on a special type of enzyme that is a total game changer for medicine. It's called the transamino. I mean, these things have some legit superpowers. They are masters at creating these things called chiral amines, which are absolutely vital building blocks for a ton of modern drugs. And they do it in a way that is way more green. They work in really gentle conditions, so they save a lot of energy, and they can replace old-school chemical processes that often need toxic and really expensive metal catalysts. And here it is. This is our protagonist. It's a class 4 transaminase originally discovered in a bacterium called Arthrobacter. This specific one was handpicked and engineered for a very special mission to completely replace a chemical step in making a pharmaceutical ingredient. All right, now for the main event. We are going to follow the incredible journey of this single enzyme, all the way from a teeny tiny sample in a lab to a massive batch in a factory. This is the scale-up story. This whole journey really happens in 3 key stages. First up is screening. You gotta sift through all the options to find the absolute best enzyme for the job. Second is lab development. This is where you perfect the recipe, but on a small scale. And third, the moment of truth, the pilot scale-up, the final test to see if this whole thing can actually work in the real world. And believe it or not, it all starts right here, in these tiny little wells. Each one holds less than a milliliter of liquid. In this specific case study we're looking at, scientists screened 10 different versions of that transaminase enzyme. Each one was a potential star. So just imagine that, after all that testing, out of 10 really promising candidates, they picked just one. The selection process is absolutely brutal. Only the top performer gets to move on to the next round. And this is where the scale just gets mind-bending. The process they perfected in those 20-liter glass jars in the lab, that's the one on the left, had to be perfectly replicated in a 10,000-liter stainless steel pilot plant. I mean, that's a 500 times jump in size. Every single variable, every temperature, every setting has to be translated perfectly. So, the million-dollar question. Did it work? Well, this chart is the answer, and it's a huge yes. The enzymatic activity, that's how well the enzyme actually performed, at the massive 10,000-liter scale was pretty much identical to what they got in the tiny 20-liter lab. I mean, what an incredible engineering success. So after going through all of that, why does this matter so much? Let's zoom back out for a second and look at the bigger picture of what this whole biocatalysis thing really means for all of us. Because at the end of that entire incredibly complex process, from a single microbe all the way up to a 10,000-liter tank, you get this, a pure, stable, super potent enzyme powder. This is the real deal, the tangible product, ready to be used in manufacturing to make life-saving drugs more efficiently, and, really importantly, more sustainably. And this quote from the researchers themselves just nails the mission. See, it's not just about making a cool enzyme. It's about fundamentally changing how we make things. It's about swapping out a piece of old-school, often harsh chemistry with a precise, elegant, and much greener biological solution. Which leaves us with one last massive thought. We've seen how this tech can revolutionize pharmaceuticals. But if we can scale up nature's tiny engines to build our medicines, what's next? What other huge global industries, like fuels, plastics, food, could these microscopic powerhouses completely rebuild from the ground up? It's pretty amazing to think about.